Good evening, everyone. I hope you enjoyed the last week's lecture on hair cycle, and I hope that now the entire process of the cyclical growth of a hair follicle from anagen to catagen to telogen, and then again going into the anagen phase of the cycle is now a bit clearer. And I've tried to make the hair cycle a lot simpler, but still comprehensive, so that it has nearly all the information that you would require minimum at the resident level and even after that. So I hope now that with the background of the hair follicle cycling, we can now understand better about the hair follicle anatomy. Now, the reason for doing the hair cycle first and then the anatomy is because I wanted to first make it easy and make it understandable what happens to the hair follicle as it grows. Okay. Now we will understand the anatomy of the hair follicle. That will make it easier. People usually do it the reverse. They first teach hair anatomy and then go for hair cycle. I think it's better to understand the holistic picture of growth of a hair follicle and then gradually go, deeper, go a bit in detail about the structure of hair. So now we have understood what exactly happens to the entire pilosebaceous unit with time. With this lecture, we'll discuss the anatomy of the hair follicle and we'll go layer by layer and discuss each layer separately. Clear? So without much further ado, let's start this week's lecture on hair follicle anatomy. Let's begin. Now we know in bioembryology, about 2 weeks back we discussed embryology of the skin, that at the bulbous hair pack stage, all 8 layers of the hair follicle and the pilosebaceous unit have formed. Okay. The hair follicle undergoes repeated cycling throughout its lifetime, and this repeated cycling of growth and then denaturement and then getting ready to enter into the anagen phase is known as the hair cycle. Now, during the hair cycle, the dermal papilla, which is the papilla, the mesenchymal condensation structure, which starts the anagen hair cycle. Okay, it starts the anagen hair cycle by stimulating the hair germ of the previous cycle to develop into the hair germ of a new cycle. So this dermal papilla, along with the matricial cells, produces hair shaft, inner root sheath and outer root sheath. Okay, so this growing hair shaft is the structure which is produced by the dermal papilla along with rapidly dividing matrix cells. Clear? The hair shaft curvature depends on the shape, thickness, and the curvature of the inner root sheath and the hair canal. So the entire structure, the inner root sheath, the hair canal, the shape of the hair shaft dictates how the hair would be straight or it would be curly. So in a normal hair shaft, you have a circular cross section of the hair shaft. So this is a hair shaft. Okay. Now, in curly hair, for example, naturally in pubic hair or eyelashes, you have that curls, and that curl is because of an elliptical cross section. An elliptical cross section, clear? And if the cross section is triangular, it is uncombable. So, in African American race, you have this kind of uncombable hair, which you call it as perm, and it becomes very difficult to you know to comb them. So in multiple media you would have seen like this kind of afro hairstyle with the African American race, and it becomes very difficult to comb, and that is because of a triangular cross section. Now in African American race, the elliptical cross section happens from the growth of the hair shaft itself. While in areas like in a Caucasian race, in sites like eyelashes, it happens when the hair is just about to exit the skin. So that is why they have curler hair or more curly hair as compared to the Caucasian races. Clear? Let's move forward. Now, this is the pilosebaceous unit. I will not go much into detail because we have covered that in the last week's lecture. I will just remind the structures of a pilosebaceous unit. Okay, so from the opening, this is the orifice. Okay, the hair canal orifice or the pilosebaceous orifice. The entire structure, the entire tube in which the hair grows, the tube, is known as the hair canal. Okay, from the opening till the opening of the sebaceous duct, it is the infundibulum. Infundibulum means funnel, so it's like a funnel. Clear? So infundibulum means funnel. From the opening of the sebaceous gland till the attachment of the erector muscle, you have the isthmus, which means the neck. From below downwards, you have the hair stem. At the attachment of the erector pili, the outer root sheath has a bulge of cell which is known as the hair bulge. Clear? So from the bulge to the downward till the dermal papilla, you have the hair stem. And below that, you have the hair bulb. Clear? It is seen in about 10 to 14 weeks embryonic gestational age. There are four classes: terminal, like on scalp and beard; apopilosebaceous, like in axilla or groin, which develop during puberty. Most other hair are vellus, and sebaceous on the sebaceous areas. Clear? Seborrheic areas. Let's move forward. Now, this table highlights the difference between terminal vellus and lanugo hair, along with another column for miniaturized. So terminal has diameter about 60 micrometer. Vellus has 30, half of it. Lanugo is somewhere in between 40. In miniaturized, the diameter is less than 30 micrometer. So even thinner than vellus hair. Okay. Now, medulla is a characteristic of terminal hair, and not all terminal hair have medulla. Some thicker hairs will have medulla, while it is absent in the other two, and may or may not be present in miniaturized hair depending on the stage of miniaturization. If it is miniaturized a lot, then medulla will not be there. The length is longer, round about 100 centimeter, while vellus, lanugo, and miniaturized are less than 2 centimeters. Clear? The hair bulb lies in the subcutaneous fat in terminal, while it lies in the reticular dermis in case of vellus hair. We know the sites of terminal hair as scalp, eyebrows, eyelashes, and all other sites are in vellus hair. Now, during puberty, vellus hair will turn into terminal hair on specific sites like the axilla or the pubic region or the beard region. Okay. While during androgenetic alopecia, the terminal hair will turn into vellus hair. Okay. The duration of anagen is also important. It is more than 1 year in terminal hair, about 1 or 2 weeks in vellus, 1 or 3 months in lanugo. We know lanugo is intrauterine, which are shed intrauterine or just after birth. Okay. But in miniaturized, the duration of anagen is reduced to less than a week. And because of this reduction, the hair doesn't grow properly. The pathology behind androgenetic alopecia. Clear? Let's move forward. With that background, this general background of pilosebaceous unit, let's understand hair anatomy now. So this is a hair follicle. It's a diagrammatic representation of a hair follicle which is not to scale. Let's enlarge it and look at the hair follicle properly. Now this large hair follicle, we are looking at the lower part of the hair follicle. Let's see the deeper layers. Starting from outside to inside, you have the connective tissue sheath. Now this connective tissue sheath is continuous with the epidermal, dermoepidermal junction and all the dermal epidermal connective tissue. All of this gets compressed and combines into a connective tissue sheath. And this sheath is what eventually forms the fibrous stela during the telogen phase. Okay, so go back and see the video on hair cycle and you will understand what is the collapse of the connective tissue sheath which leads to the formation of fibrous stela or fibrous streamer. The next layer is outer root sheath. We'll discuss each layer separately, don't worry. Now we have the connective tissue sheath, we have the ORS. Now we have the companion layer, the Henle layer. Henle is outside, the Huxley layer, which is inside, and the IRS cuticle, inner root sheath cuticle. All four together are known as the inner root sheath. Clear? So parts of IRS is IRS cuticle, Huxley layer, Henle layer, and companion layer from inside to outside. Clear? Now let's move forward. The hair cuticle, which is a bit separate from the IRS cuticle, along with the cortex and the medulla together forms the hair shaft. And this hair shaft is the hair which eventually comes out of the follicle or the opening, the pore or the orifice. Clear? So again, recapping, from outside to inside, we have the connective tissue sheath, then we have the ORS, then we have companion layer, Henle layer, Huxley layer, and IRS cuticle. All four of these together form inner root sheath. Then we have hair cuticle, cortex, and medulla, which together form the hair shaft. We are clear on that? So we have looked at the longitudinal section of a hair follicle. Let's look at the transverse section. So this is a schematic representation of the transverse section of a hair follicle, and this is just, you could say, above the bulb. And you can see iris, that means it is below the isthmus. So in between the isthmus and the bulb region, or you could say the hair stem. So we have taken a section at that level. So from outside to inside, we have the connective tissue sheath. Then we have the ORS, and you can look at that the majority bulk of the perifollicular unit is made by the ORS. That's why it's the thickest layer of that unit. Clear? As we go inside, we have the iris cure. We have the companion layer, we have the Henle layer, we have the Huxley layer, and the iris cuticle together forming the inner root sheath. So this IRS plus ORS plus connective tissue sheath is part of the perifollicular unit. Clear? Now we talk about the hair shaft. We have the hair cuticle, then we have the cortex, and in the center we have the medulla. Now bulk of the hair follicle is formed by the cortex. This is the thickest layer of the hair shaft. So cortex is the thickest layer of the hair shaft, while the ORS is the thickest layer of the perifollicular unit. Clear? Let's move forward. So let's talk about each and individual layer separately, and I'll be using the schematic representation of the pilosebaceous unit. Clear? So let's talk about the outer root sheath. Outer root sheath is continuous with the epidermis of the skin at the level of infundibulum. So infundibulum ends at the attachment or the wherever the duct for the sebaceous gland pours into the follicle. So at that level you have the infundibulum from the opening to the attachment of sebaceous gland. Clear? At the attachment sebaceous gland, ORS becomes continuous with the epidermis. Clear? Below that, there is a point where the inner root sheath, here it is shown in purple, the inner root sheath gets separated from the ORS. Clear? After the IRS has separated from the ORS, any keratinization above that level, okay, above that level will be epidermal. That means this keratinization will have a granular layer. So if you look at the longitudinal section of a hair follicle, any part, the part of ORS which is above the IRS has sloughed off, you will see epidermal keratinization. That means you will have granular layer. That means you will have keratohyalin granules that contain keratins specific for skin keratinization, not hair keratinization. Clear? Below that level, you will have keratinization which is known as tricholemmal keratinization. Remember, another name for ORS is tricholemma. Okay, it's another name for ORS is tricholemma. So below that attachment, below that level, you have tricholemmal keratinization. You will not have a granular layer. The granules will be present in the keratinocyte, but they will have keratins specific for hair. If you want to know more about keratins, see our video on basics of keratinization. I've talked about what are skin keratins and what are hair keratins. Okay. Now, the part of ORS where the erector pili attaches, it bulges out. It is thickened, and that is known as hair bulge. And this houses the stem cells. So all these stem cells are there at the hair bulge. And these stem cells are responsible for creating a new follicle every time the follicle enters into the anagen phase. Clear? Now this bulge is thickest at the suprabulbar region. The thickening is known as the bulge. And at the hair bulb, that means just below, the ORS, the outer root sheath, goes below the hair canal and engulfs the dermal papilla. So you can see a little bit here that it is going around and cupping the dermal papilla, and at this stage it is single layer at the hair bulb stage. Clear? Let's move forward. So now what we have done is we have expanded the lower part and we have removed the outer root sheath. We are just looking at the inner root sheath now. Okay. So the inner root sheath exists from the base till the isthmus. And after isthmus, it is destroyed by the proteolytic enzymes. So you have the level at which it ends is roughly the isthmus. Now, IRS is made by the matrix proliferating cells. We have told you that dermal papilla with matricial cells will form IRS and the hair shaft and ORS. Okay. Now, inner root sheath has four layers. From outside to inside, you have the companion layer, Henle layer, Huxley layer, and the IRS cuticle. So let's look at these layers. So you have the outside, you have the companion layer, then you have the Henle layer, Huxley, and IRS cuticle. The first layer that we'll discuss will be companion layer. Now, companion layer is a part of IRS, inner root sheath. Earlier, it was considered a part of outer root sheath, but it has now been shown to be a part of inner root sheath. It is a single layer of flat cells. The cells are flat and single layer. You can see in the schematic diagram, it is flat. Now, this companion layer helps in the gliding of inner root sheath against the outer root sheath. So, this whole part is IRS. This whole part is also IRS. And in between you have the hair shaft. Clear? It's a longitudinal section. Now outside that lies the ORS. Clear? So as the hair grows, it carries the inner root sheath along with itself, and inner root sheath is growing from inside to outside, so it glides on the ORS, and this gliding is helped by the companion layer. That is why this plane at which the gliding is happening is known as gliding or slippage plane, and that is formed by the companion layer. It has specific hair keratin K6HF type 2. Okay. So this helps in the movement of IRS over ORS as the hair shaft grows outside the skin. The second layer is the Huxley layer. So Huxley layer is just you could say just above the IRS cuticle. Okay. So you might get confused why we are discussing Huxley first and Henle later, because I think Huxley is slightly more important than Henle, and it is thicker also. So it's important to discuss Huxley, but don't get confused. From outside to inside, it is companion layer, Henle layer, Huxley layer, and then you have the cuticle, iris cuticle. Now, Huxley layer is 2 to 4 celled layer, so it's thicker, and it keratinizes above Adamson fringe. Now, what is Adamson fringe? We'll discuss. If you're a member of the channel, you will have a separate discussion on Adamson fringe at the end. Now, there are certain cells known as Flugel-Zillen cells or wing cells. These cells are from the Huxley layer. They're part of the Huxley layer, but they go inside the Henle layer, through the Henle layer, and attach to the companion layer. So these are known as wing cells. They spread like wings. The Flugel-Zillen or Flugel-Zillen, German term, cells. So you can see wing cells here. It's part of the Huxley layer and going through the Henle layer. The red one is the Henle layer. It goes through and attaches to the companion cell. Now, iris cuticle overlaps with the hair shaft cuticle. Remember, hair shaft cuticle is different. It's part of the hair shaft. And both of them attaches in a shingle roof appearance. So this is a shingle roof appearance. I'll attach a photograph of a shingle roof here so that you can understand what is shingle. So this kind of jagged attachment of the iris cuticle with the hair shaft cuticle provides adhesion. And that is why when the hair shaft grows, it carries the iris with it. Okay. GTH3 gene is very important for formation of Huxley layer, and Huxley layer is very important to regulate hair shaft shape. Remember, hair shaft shape, many factors contribute to it. One of the factors, IRS, and Huxley being the thickest layer, contributes a lot. Let's move forward. Now, the layer just below the companion layer is the Henle layer, the second layer. It's a single cell layer, so you can see it is how thin it is. And Henle layer is the first to develop keratohyalin granule, and it is the first to keratinize. So the first structure that starts keratinization, that hard keratinization, I should write it should be the first to keratinize by using the hard keratinization process, and because of that it becomes the first structure to show keratinization. Why? It is the first structure to form keratohyalin granules. Clear? So this could be a very good viva question or a quiz question. First structure to keratinize is Henle's layer. So I'll be using Henle's layer and Henle layer interchangeably. Just don't mind. Clear? Let's move forward. Hmm. I have removed in this schematic diagram, I've removed the iris. Now we are discussing only the hair shaft. Clear? Now hair shaft forms from the rapidly dividing matrix proliferating cells. We know that. The tip of the bulb gives rise to the cuticle, cortex, and the medulla, all three layers of the hair shaft. There exists a zone of keratinization. Here it is shown here, a zone of keratinization or a pre-keratogenous zone. And in this zone, the hard keratins start to get expressed by the action of LEF1 and BMP receptor 1A gene. Now, what exactly do we mean by that? When the matricial cells are dividing, it is just forming all the keratinocytes. And as the hair grows, different keratins get expressed. Some are hard, some are softer. Some are softer like the skin, some are hard like the hair. So, in the zone of keratinization, the expression of hard keratins increase, and the hair shaft after the zone of keratinization is formed majorly by hard keratin. Okay. The uppermost part of the zone of keratinization is an inverted V-shaped area known as the Adamson's fringe, and that marks the end of the keratinization zone. After that, the hair shaft will be keratinized. Clear? So let's move forward. If you have any doubts, mention in the comment section. Now, as the hair shaft grows, the lumen, the central part, can undergo necrosis and different cornification of epithelial cells forming the medulla. Okay. We have discussed that the curved hair arises from the curved hair follicle, majorly by the inner root sheath. If it is circular, then it is the normal hair. If it is elliptical, then it is curly. And if it is triangular, then it is uncombable. So if we look at the structures of the hair shaft, you have the dermal papilla. Overlying that, you have the matricial cells or the matrix cells, which will divide and form the hair shaft and iris. Okay. And this is the zone of keratinization. Above that ends with the Adamson's area or the Adamson's fringe. And then you have the cortex, cuticle, and medulla, the parts of hair shaft. Factors like signaling pathways like WNT required for hair growth, BMP down regulation is required for hair growth, EDAR, FGF, hedgehog, sonic hedgehog proteins are required for growth. Insulin growth factors, notch signaling, we have discussed some of them in the hair cycling video. Let's move forward. Let's discuss about the cuticle. Now, cuticle tightly wraps around the hair shaft and impacts the appearance and structure of the hair shaft. So most of the time, the shininess of the hair usually comes from the smoothness of the cuticle. Okay, so when the hair gets older, it becomes entangled and dry. It is because of the denaturement of the cuticle. It undergoes weathering as hair exits the skin. So as the hair exits, exits the skin, this cuticle undergoes weathering. It weathers off. Sometimes when the inflammatory process is you could say high, or you could say that if the proper follicular keratinization is not happening, you might see it as perifollicular scales. This cuticle as perifollicular scales, or actually a part of perifollicular scales. Clear? So this is the importance of cuticle. It tightly wraps and impacts the appearance and structure of the hair shaft. Now, the cortex forms the bulk of the hair shaft. The major part of the hair shaft is made by the cortex. In the cortex, you have melanin. So, last week when we were discussing the hair cycle, we discussed that the pigmentation is transferred only to the cortical cells by the hair melanocytes. So, cortex is the structure which is melanized. Cortex consists of larger cable-like structures like macrofibrils, and when you zoom in, or when you go more microscopically, you will see that macrofibrils will have intermediate filaments, and we know from our knowledge of keratinization that intermediate filaments are made by the keratin molecules. Okay, if you want to revise it, or if you want to know more about intermediate filaments and keratinization, look at our video on basics of keratinization. Clear? Let's move forward. Now, medulla. Medulla is found in the center of larger follicles. So the follicle, the hair has to be thick to have a substantial medulla. The keratin expression is different from what we get in the cortex, and usually the areas which are androgen dependent, like the beard or the axilla, or in fact the scalp in AGA, the keratins which are under the control of androgens are mostly found in the medulla. Clear? Let's move forward. So now what has happened? We have enlarged the lower part of the hair shaft, and now we'll discuss the dermal papilla. Okay, so you can see the structure here. You have the dermal papilla, which is actually a core of dermal tissue, a condensation of dermal tissue. And this dermal tissue is specially organized to create a new follicle, or at least provide signals for the growth of new follicle. Now, this dermal tissue has a cap on it, and this cap is formed by the matrix cells. So here the matricial cells or the matrix cells are shown in purple. It's like a cap. In between the core of the dermal papilla, you have the capillary loop. Every dermal papilla has a single capillary loop for nutrition. Okay. Inside the dermal papilla, you have all the extracellular matrix. You have collagen fibers that provide structure. Okay. Now, dermal papilla is also responsible for the formation of perifollicular sheath. And this perifollicular sheath goes around, encircles the ORS and becomes the connective tissue sheath around the follicle. Clear? Dermal papilla is a core of mesenchymally derived tissue enveloped by the matrix epithelium. So it is enveloped like a cap. So all the fibroblasts, collagen bundles, mucopolysaccharide stroma, nerve fibers, single capillary loop are all in the dermal papilla. And dermal papilla is continuous with the perifollicular sheath that envelops the lower follicle. That is why perifollicular sheath is also known as dermal sheath. Clear? Let's move forward. Now, the volume of dermal papilla, that means the amount of dermal dividing cells and the matrix cells, the volume is directly proportional to the number of matrix cells. Now, remember, if the core is large, the covering is also large, and the covering is made by the matrix cells. So, the matrix cells are more in number, the thicker will be the hair shaft because the amount of keratinocytes that they are producing will be a lot more. Okay. Now, dermal papilla induces hair follicle generation. We know it starts the anagen phase by moving upwards and inducing the secondary hair germ. If you want to go into more detail, look at last week's lecture on hair cycle. It produces all sorts of growth factors like keratinocyte growth factor and FGF receptor 2, all a lot of growth factors which is responsible for the cycling of the hair follicle. Okay, so let's move forward. Now the matricial cells. These are germinative cells. These are also known as germinative cells. They have a very high rate of division. In fact, they have one of the highest rate of division in the entire body. Okay. We know that the volume and the number of matrix cells is directly proportional to the size of the hair shaft. They are composed of spindle-shaped central cortex cells, which will give rise to the cortex of the hair shaft. An outer layer of flattened overlapping cuticle cells will give rise to the hair shaft cuticle. So you have the cortex, the surrounding area, or you could say that roughly this part will give rise to the cuticle, all the other part will give rise to the cortex, while the central tip part will give rise to the medulla. Okay, let's move forward. So we'll discuss a bit about the perifollicular sheath. The perifollicular sheath envelops the epithelial components of the hair follicle. Okay, it has an inner basement membrane. In the inside, it is basement membrane, which is also known as vitreous or glassy membrane. Remember the importance of glassy membrane during the catagen phase. Look at last week's video. And the outer connective tissue sheath. Clear? Now, follicular basement membrane is contiguous with the epidermal basement membrane. In all the diagrams of pilosebaceous unit, you will see that the basement membrane of the hair follicle goes upwards and is continuous with the skin basement membrane. Clear? During catagen, basement membrane thickens and then disintegrates, and this disintegration pulls the dermal papilla along with it so that the next anagen phase can start. The major collagen is type 3 collagen, which forms the connective tissue sheath. In the upper follicle, this connective tissue sheath is continuous with the surrounding papillary dermis. So if you look at a very simplified diagram of a follicle, in the upper follicle, the perifollicular sheath is continuous with the skin, the papillary dermis. While below that, you have two types of fibers. Inner, you have the circular fiber, and these circular fibers engulf it. Okay, it goes, if this is the follicle, it goes around the follicle and wraps it. So it's a circular. While the outer layer is longitudinal, so it is in this direction, longitudinal. Okay, so inner circular, outside longitudinally, it wraps around the lower follicle. Now, there have been instances where perifollicular sheath can form a new follicle. In fact, there was one study in which a part of perifollicular sheath was transplanted to a non-follicle developing region like palms and soles and still tried to develop the hair follicle. But remember, the major chunk of hair follicle generation happens from the dermal papilla and the matricial cells. Clear? But perifollicular sheath can act as a source of mesenchymal stem cells. It's an important source of mesenchymal stem cells, and this can be a very unique opportunity to act as a source of wound repair. So whenever hair is plucked, it is acted as a wound, and in fact there have been instances where transplanted hair has generated wound repair in a damaged skin part, damaged skin tissue. Okay, so let's move forward. Now, so recapping the hair follicle layers, and this diagram I've taken from Bologna. So from below to upwards, if you take a section at the hair bulb, you have in the center, you have the dermal papilla. Let me just change the pen color. In the center, you have the dermal papilla. Then you have the MC is hair matrix cells. These are the matricial layer responsible for growth. Outside, you have the ORS. The orange is the IRS. And then you have all the perifollicular structures. As you go upwards, you will gradually see that a dermal papilla has gone. It is replaced by the hair shaft, which is produced by the dermal papilla. Gradually, at the level of isthmus, the iris is gone. So you can look at here that at the isthmus level, you don't see much of iris. You will see on the hair shaft and ORS. And the ORS is contiguous with the papillary layer. So as we move upwards, you can see that a gap is being made in between the hair shaft and the ORS. And why is there a gap between the hair shaft and the ORS? Because the IRS has disintegrated. Okay, and at the level at which the IRS gets disintegrated, above that, the ORS will keratinize by epidermal keratinization, while below that, the keratinization will be tricholemmal. And that is how you differentiate between epidermoid cyst and a tricholemmal cyst. You look at the layer of the cyst and see if you can find the granular layer and the the hyaline granules. If the granules are present, if the granular layer is present, it is epidermal keratinization. That means the cyst is formed from structures above the level of isthmus, so it's more likely an epidermoid cyst. If you don't see the granular layer, that means it has underwent tricholemmal keratinization. So it is below the isthmus, and you have a tricholemmal cyst. Clear? Let's move forward. So here is a complete amalgamation of all the structures that we have talked about, and you can actually see the single capillary loop in the dermal papilla. You can see it here, and it is part from the perifollicular arteries or the plexus of arteries, and you can also see the perifollicular neural network and the erector muscles, erector pili muscle, and the hair shaft. Clear? This is longitudinal section. Earlier we saw the stages or different levels of cuts. Let's move forward. Now, functions of hair and nail, I decided to cover. It will hardly take a minute, but this table from Bologna actually highlights, is more or less exhaustive of all the functions of hair. So for hair shaft, it is for communication, camouflage, any chemical or physical damage, it prevents it. It prevents against insects. It provides sensory antenna, so you can somehow sense when something is moving. Insulation against heat loss, overheating, or dryness. Removal from the skin surface of parasites, so you can just brush them off. And in fact, some parasites can get entangled in the hair. And this is one of the functions of eyebrows and eyelashes. It traps the dust particles and prevent it from going into the eye. In fact, eyebrows prevent the forehead sweat from going into the eye. Okay, facilitation of sebum and sweat. That is, see, the perifollicular unit is the opening for eccrine sweat. for sebum, sorry, not for eccrine, for apocrine sweat and sebum. So all of these secretions go outside from the hair canal, and while they are secreted inside the hair canal, the growth of the hair shaft pulls the structures above by capillary action, and that is how these secretions go outside the skin. Okay. So let's come to the functional properties of pilosebaceous unit. It is, you could say, the attachment of the eccrine gland and the sebaceous gland gives rise to all the different secretions. The pilosebaceous unit can individually act as a reservoir of stem cells, as a reservoir for Langerhans cells, as a reservoir for Merkel cells, keratinocytes, melanocytes. All of this can, all the stem cells that can create this different lineages of cells are present in a pilosebaceous unit. Clear? So let's move forward. So with that, we finish our lecture on hair anatomy. These are the references for the chapters in individual textbooks. I've utilized different articles. Individual references are there on the slide itself. But if you've gone through this lecture, I hope and sincerely wish that your clarity about the hair follicle structure, its anatomy, is now clear. Combine this video with the last week's video of hair cycling, and you have nearly everything you need to know about a hair follicle functioning, normal function, anatomy, and physiology. Clear? With that, again, shameless plug. This is the last week. It's available at 59 a month. The membership for the channel will be there for about a few more days, 2-3 days, till 31st of December. After that, the rates will be increased. And those who have become member of the channel should share this information to other people so that they can also come. You can copy paste the link, it's there in the description. And the videos which are there for the members only are much more detailed, structured, and there were a lot of different things. Okay, multiple times I have to discuss clinical cases which I cannot discuss as per the YouTube guidelines, but for members I can freely discuss colored photographs and photographs which I would not be that comfortable sharing in a public platform. So it's better to get a membership. And of course, if any new topics are asked, the importance are first given to the members. So if you have gone through any of my videos before, or this video perhaps, and if you think that this video is actually helping you during residency, or just understanding the basic concept better, do consider becoming a member of the channel. Okay, so discounted rates available till the new year. So with that, this would be the last video of 2024. We'll meet again next year in the new year in 2025. We'll continue our series. I'll try to bring a video every week, and I will not waste much time. I'm taking this opportunity to wish every one of you a very happy and prosperous new year. May this new year be a lot better than what has gone by, and a lesser of the negative things that you have underwent this year. More of positive vibes, more of positive aspects. A very, very happy new year. Sincerest wishes. May this year be blissful. You remain healthy and prosperous. With that, I'll finish this lecture. I will not take much time. The year is ending. So is this lecture. We will not waste time. Adios and bye bye.